I want to give an overview of what the 5P medicine framework is and where multiomics fits in there. We will take a look at especially the place of multiomics, of metabolomics, sorry, in multiomics. Then I will look at what 5P medicine is, and we will go through several case studies to understand what are the different connection points where metabolomics enables 5P medicine, and often that will be in conjunction with other omics, so already in multiomics studies. And we will conclude with the way forward with multiomics, whether you're familiar with that or not, like how can you already start employing metabolomics in your multiomics approaches and applications. So the first topic is what is multiomics and what is the place of metabolomics in there. I always like to begin with a kind of high-level picture of this because my background is in multiomics, actually. I did this for nearly 10 years before joining biocrates. And I always found it fascinating how we've kind of created a problem for ourselves in the sense that when we took a cell or an organism or any matrix that has DNAs, RNAs, proteins, metabolites interacting with each other, we wanted to look at that more closely. And so we've separated those molecules based on physicochemical properties. When we did that, we created beautifully high-resolution pictures that we call omics, like omic profiles, for example, of one of these types of molecules. And we can check, comparing, for example, case control or treatment versus control, differences between different conditions. But of course, we lose the complexity of the biological system when we do this. So multiomics is our way of kind of moving back from this reductionist or simplified way of looking at biology into adding complexity to it by assembling those different omics. And to do that, basically, everyone is using their best guess at how to combine those things together. And it's always going to be a mixture of bioinformatics and more machine learning algorithms, and also data interpretation, which can be a much more hands-on work based on biological knowledge. And so the point I want to put through here is that we have created a problem for ourselves, but for some of us, that creates a very interesting puzzle that we have to solve, that is this multiomics integration, which can be really fun work to do. And so what is the place of metabolomics in that puzzle? What does it bring? And in general, like people will usually come to a biological problem from the point of view of which omics should I use rather than I have metabolomics or genomics and what should I apply it to? And so, of course, you have choices between these different pieces of puzzles. And I really see genomics and metabolomics as like two very complementary pieces. And then the other pieces of the puzzle that can really bring very interesting add-ons as well. So if we start from genomics, which is the omic that most people will be familiar with because it's the one that's been there the longest, it's a very static form of biology. So it's the genomic code that is going to be responsible for which RNAs and proteins are transcribed and translated. But it's very static. So you have your genome at birth, and then more or less there are not going to be too many changes. That's convenient for statistical analysis because an individual is going to have one profile. But it's going to inform on genetic predisposition, especially if only one or few genes are responsible for a disease or for a high risk of disease. But when you come to diseases such as complex chronic disease like autoimmune disease or certain forms of cancer or a certain form of neurological, for example, degenerative disease, we know that the way we live our life is going to have a huge impact on the risk of an individual to actually develop the disease. So for that, genomics is only going to provide us a fraction of the picture, usually 10 to 20% of the variance that exists in the population. And the other omics are going to be much better at reflecting the impact of how we live our lives. So that's what we eat, how stressed we are, how we sleep, the chemicals we're exposed to, all those things, the drugs we take, they all influence our biology, and they can all make us more susceptible to disease, but they can also make us more resistant to disease. So monitoring that more flexible aspect of biology can be a very positive thing too, but you have to have the right tools to do it. And this is where metabolomics comes in. Metabolomics is a highly sensitive omic to diet, to other omics, to the influence of other omics, and also to exposures and all sorts of factors that are going to influence our health acutely, but also chronically. So that's why a couple of years ago, when we wrote our white paper on complex chronic diseases and their common origin, especially in diet, we looked at that through the lens of the metabolome, and we looked at the effect of food on the microbiome and the combined effect on metabolites and how they are processed in our body. So if you're interested in Alzheimer's disease, in cancer, in autoimmune disease, I encourage you to look into this white paper. And but we will see lots of other examples in the rest of the talk where you're going to get some inspiration, hopefully, to not only use metabolomics, but also use metabolomics in a multiomic context. And so the first example I have here is a case study of like what's the kind of multiomic integration I did for many years. So this is not a study where I contributed, but it's a similar approach to what I did in the past, where you have your different omics and you're going to analyze each omic individually, and then you're going to try and combine them to get more than the sum of its parts. So what we see here is a study on a relatively few cases of Parkinson's disease. So 40 individuals with Parkinson's disease versus 30 individuals that are considered controls. And what we're looking at here is DNA methylation arrays and metabolomics in the brains of these patients post-mortem. So what are the differences between Parkinson's disease and control patients? So what you see on the left is a volcano plot of the metabolomic data, and you see in orange the metabolites that are higher in the Parkinson's disease brains, and in blue the ones that are in lower abundance in those brains compared to control. And you'll notice that taurine, which is kind of in the middle, I should go here, maybe you see my mouse, taurine that is kind of in the middle of the orange dots, is there, but is not necessarily the one that you're going to look at first. But then when you overlay that and you look for associations with the epigenomic data set, this is where taurine actually becomes really relevant, because that is the one analyte that you see on the left of the circus plot that is associated with a number of loci that are encoding proteins that have to do with bile acid metabolism. So for anyone who knows about taurine, you know that this is a metabolite that is often conjugated to bile acids, so it has a direct metabolic link to bile acids. And interestingly, this is the one that actually associated with those genes also related to bile acid metabolism. So this is a simple but very powerful example for me of how simply combining two layers, and if you have more layers to play with, go ahead, like combine more omic layers together to try and figure out in each data set which aspects are going to be more interesting and where the pathways kind of cross across those different layers. So this was a couple of examples, really high level of multiomics and metabolomics, but so I want to put this into the context that I promised you, which is 5P medicine. And this is also going to get us closer to the topics that are going to be discussed by our next speakers. So 5P medicine is a concept that I didn't invent. This is something that has been around for a few years already. A few years ago, it was 3P, then it became 4P, and now you can look even at 5P. And so it's basically a framework to make it easier to understand the different places in the future developments of medicine where omics are going to play a part. It's not exclusive to metabolomics. It's really a kind of inclusive framework where all omics can feed into. And so those 5Ps you see represented here in blue, preventive, predictive, precision medicine, population-based medicine, and participatory medicine. And in the next slides, I will take you through a few case studies where we're going to go through 4 of those Ps. I will leave out the participatory medicine for this talk because I simply don't have enough time, but participatory medicine is a really, really relevant one, especially when we start to have the right tools that have been developed in the other parts, then people can really take things more in their own hands, and we can have a much more proactive type of medicine that is driven even by the patient. And I think Sabine Ban will give us a good example of how also the patients are going to be able to also contribute to what happens there. So I will focus on the other 4 Ps, and each time we're going to look at the relevance of metabolomics for this P and some examples. So of course, we start with prevention and preventive medicine. This makes sense chronologically because if you can prevent disease, you're not going to need biomarkers, you're not going to need any precision medicine, you're actually going to prevent the onset of disease, which is why I tend to say that this is my favorite P. If we manage to get that one down, we can remove a lot of work down the line after we've taken care of the people who are already sick. But so in terms of preventive medicine, metabolomics is going to feed in there really well. So we all know about the polygenetic risk scores. You can also calculate metabolomics risk scores. And with that, you can start to have an idea whether people have a higher risk of developing a disease based on their lifestyle, on how they live their life. When you know um, what makes them more likely to have a disease occur, you can also have better ideas of which interventions should be applied. That can be therapeutic, that can be dietary intervention, that can be lifestyle intervention. Often it's going to be a combination of all of those. And then you can still use metabolomics then to monitor, not only when people start to get in the red zone, but also to monitor if they get back to what we would consider a normal phenotype. So there are lots of potential applications. And I want to show you here one example that has to do with lipidomics that I discussed recently on my podcast, The Metabolomist, with Christina Lerido Quigley. And this is from a study that she and her group put together with obese and overweight children and adolescents. So the study had over 1,000 individuals, where the first analysis was a simple comparison, so case control, comparing the lipidomic profile of obese children versus non-obese children. If you know your lipid classes, you will recognize from the bottom up some classes you would expect. So triglycerides at the very bottom, for example, in orange, you expect those to be higher in people with obesity or in children with obesity. And then for glycerophospholipid, we have a mix of higher and lower species. And for sphingolipids, also a bunch of especially sphingomyelids and ceramides that seem to be quite high. So this is not surprising. This is a profile that is expected. But what's really interesting about this study is the second part. In the second part, they did a longitudinal study where they applied, so tailored dietary and lifestyle intervention for 200 of these children. And then they let them follow those interventions for a year and they measured again the lipidome. And here you see the changes that are associated with the changes in BMI. You see the triglycerides drastically reduced. You see the glycerophospholipids are changed and the sphingolipids are changed. So all those markers that were associated with obesity in terms of the lipids in the blood, they were strongly influenced in the children where the intervention worked. So that's a strong a sign that you can really monitor these changes, whether it's lipidomics or metabolomics. I often associate the two, but in this case for lipidomics, it worked really, really well. If we go now to predictive medicine, of course, the first thing that comes to mind is biomarkers. And this is what a lot of people think metabolomics is about, is finding biomarkers. And again, we will see beautiful examples of this in the two talks that are coming. But you will see that for each of these talks, there is much more than prediction that is actually going to be down the road of those studies. Because when you the beauty of using omics is that you don't only get data points, you also get data points that have functions associated to them. And this is going to help you to better understand the disease, to think of better treatment options. And this is where it starts to be really, really interesting in terms of progressing how we tackle disease. One place where biomarkers are often searched for is either early biomarkers. We will hear about this in the talk by Oliver Bathe on colorectal cancer, but also drug response prediction. And so the example that I'm not going to discuss, because it's going to be discussed much better by one of our guest speakers, is an example of biomarker signature to differentiate, so to give better diagnostics for bipolar versus unipolar depression. This is also something that I discussed with Sabine Ban on the podcast, but I will let her discuss this. But needless to say, that is going to be a very interesting talk where you're going to learn about all the strategy of how they built that study and the very interesting markers that they found. Precision medicine is the third P. This is probably the most popular one and the most buzzword P of the 5P. And what happens there with metabolomics and other omics is most often a type of stratification. With metabolomics, we call that metabotyping. So you're going to use your broad profiling tool to kind of break down a sort of monolithic diagnostic, for example, into subgroups. And then each of those subgroups you can address with personalized approaches, so treatment or intervention and so on. This is the example I have here from a discussion with Rachel Kelly. This is another episode of the podcast. And what you see in the box plots on the left, you see the top four different measures of lung function and 5 subgroups of a broader group of children with asthma that are separated based on their metabotype, so based on their kind of stratification profile. We have those 5 subgroups. They have different responses to the different tests of lung function. And on top, you see the first cohort, and on the bottom, you see a second validation cohort from a different continent, where those ways of defining the subgroups really hold well in a different population. And then once they had these subgroups and they knew which metabolites and lipids were defining these subgroups, they could start to look at the mechanistic differences between them. And so the idea here is, again, like finding not just better subgroup definition where each might have a different kind of adapted treatment, but also better understanding maybe the etiology or at least the molecular presentation of the disease in these different subgroups. This takes us to the last P that I will talk about today, population-based medicine. You remember I was telling you about the genetic predisposition at the beginning. So the other side of this, so the genetic variance, the other side of this would be the environmental variance, where metabolomics and other omics can really add to genomics to kind of look at how the exposures, the habits, the lifestyle of the different parts of the population will influence different subphenotypes. And I put multiomics here. So multiomics you can use in any one of those P's, but the one where it's most used at the moment is most probably population-based medicine, because this is typically an environment where you're looking for higher endpoints, higher numbers of data points, because you're working with large number of patients. And so the example we will look at here is a cohort called the Alpine CRIS cohort from Italy. And here we had over 3,000 individuals where whole exome sequencing was measured as well as metabolomics. And here they did the typical game of looking for associations between the metabolites and the genes, the gene variants that are present in the population. So this is also done in MGWAS, for example, where you're going to have genome-wide association studies, and you're going to combine that with the metabolome to have this new type of trait that you look for association with. Here it was done with this whole exome sequencing, and they found 85 associations between variant and trait, including 39 that were not known before. And so among those 39, there were some that were, as is often the case, associations between genes and metabolites where you see a clear functional or mechanistic relationship between them. So typically like an enzyme and its substrate, or things like a transporter and what it transports, things like this. So this is also something that gives you an idea that, yes, there are implications of the genetic variability into the metabolome, but sometimes you're going to also find things that are a bit more remote, where it takes a bit more work to figure out what the connection is. And here we have a multiomic study focused on Alzheimer's. So this is a study of over 1,500 individuals. And what was looked at here is genetics, transcriptomics, proteomics, and metabolomics. So really the whole toolbox of the typical toolbox of multiomics. And here we see associations between, for example, transcriptomics and metabolomics are quite strong. We see certain markers. If we look at the bar charts, the red, green, and blue bar charts, you see association of different levels of cognitive impairment with certain types of metabolites. So here, certain types of acylcarnitines, but also with certain proteins. And so putting all those things together, this is where you can start to play the fun game of trying to interpret what the associations you find actually mean. So you can start to not only find biomarkers, of course, because sometimes you just want to find something that is kind of a sensor for what you're looking at, but also to understand why that specific metabolite or why that specific protein is actually relevant in the disease context you're looking at. So I hope that these examples made you a bit excited to try, of course, metabolomics for 5P medicine and for the different applications that really ring a bell for you, but also for using multiomics. And I know many of us don't necessarily know how to do that, or they're not bioinformaticians and they're not bioinformatics savvy. So for that reason, we also wanted to make it easy for people to make that step. When you think of the way of approaching multiomics analysis, I would say there are two main ways. So either you have a static piece of software that has pre-made sets of analyses and you're going to throw your data at it. And usually you're going to spend some time also like polishing your data so it's a good fit for that software. But eventually when it works, you can just click a few buttons and have a predefined set of analyses that you can run. But it's never going to be really tailored to your study. The complete flip side of that is having your own bioinformatician, which some of us have, but unfortunately not everyone has yet. But when you do have that person and they know all different types of omic data sets, then it's really easy to have a tailored approach for each study. What we're proposing at biocrates is something that's at the interface of those two, where we have teamed up with a team of bioinformaticians who have semi-automated pipelines for all sorts of omics. And then they run those pipelines. They're very tailored to the type of omic, and then they do the integration together. It's very quick, but it's also still, there's always a bioinformatician that checks the workflow, that checks that it makes sense for the study design, and this is what we liked a lot about it. So the reports look something like this, depending on the different omics you have. You're going to have, of course, a different layout. But so basically all sorts of omics are covered by this and can be integrated together. And there's always at the end a layer of like integration, which we can also add an additional layer to. If we want to do data interpretation, we can also do that on top. So like really the interpretation that is made by a human. The added value of this is you get transparent bioinformatics, which I think is really important. Also that you have results that you not only you can understand, but that you can also directly use in publications or also follow up on afterwards. You get advanced interpretation if you choose to. So you can really get like the biological, the kind of really solving the puzzle to the very end on top. And the turnaround time is really quick. It's really a matter of a few weeks. So the question for each of you at the end of this talk for me is, what's the best combination of omics for me? Well, that really depends on your goals. So many of you probably know I wrote that book a couple of years ago called The Story Principle, that's focused on metabolomics and on really like defining your research question, finding the right tools to approach it. And I think multiomics is exactly the same. So what is the omic that is going to provide me with the information that I need to actually answer my questions? And then who do I work with to get this done? And how do I combine those things together? If you don't know the story principle, I encourage you to take a look, but this can just give you a little bit an idea of where to start as well. So that is it for me today. We still have time for a couple of questions. If you want to check out our multiomic data analysis service, this is the information there. You find it on our website. And if you want to learn more about metabolomics, I encourage you to stay curious.